Welcome back to the Explainer. There's this one question that I think keeps every researcher up at night, and that is, can I really trust my qPCR data? Today we're digging into Bio-Rad's CFX Maestro software, and we're going to unlock two super powerful tools that will help you get those publication-ready results every single time.
Okay, let's just be real for a second. You've been there, right? Staring at that spreadsheet just full of CQ values, and you get that little pang of doubt in your stomach. Is this data actually clean? Or is there some hidden flaw that could totally mess up my final conclusion?
So before we get to the cool solutions, we got to talk about the culprits. There are two major hurdles, and they're often hiding right in plain sight, just waiting to stand between you and data you can actually trust.
It really is a classic story, isn't it? On one side, we've got our goal, beautiful, clean, reproducible data that we can proudly publish. But on the other side, two huge pitfalls are just waiting to trip us up: bad normalization and missed quality control failures. And if either one of those goes wrong, well, the integrity of your entire experiment is on the line.
So let's break down that first hurdle, normalization. The best way to think of it is like a race. It corrects for all those tiny little variations between your samples, like maybe you pipetted a tiny bit more material in one well. It makes sure every single runner starts from the exact same line. Without that, you just can't tell if a result is real or if it's just a technical hiccup.
Okay, so how do we nail that normalization? Well, the first tool we're going to look at is the reference gene selection tool. And honestly, it's a total game changer for picking your internal controls. You're going to love this.
No more just picking the usual housekeeping genes and just crossing your fingers, right? This tool does all the heavy lifting for you. It systematically looks at up to 30 of your potential reference genes across all of your samples to find the ones that are absolutely rock solid and the most reliable for your specific experiment.
So the software does all this amazing work, but you still got to know what you're looking at, right? Well, luckily, it lays out all the data in this super clear visual way that makes your decision incredibly easy.
And this is the best part. It is so simple. The software gives you this bar chart, and all you have to do is look for the tallest bars. A higher stability value just means that gene's expression level doesn't bounce around all over the place between your samples. It's consistent, and that makes it the perfect anchor for your normalization.
And it gets even better. They've built in this, it's basically a traffic light system. It's genius. Genes in green, those are your gold standard. Use them. The yellow ones are okay, they're acceptable, but you'll probably want to combine a few just to be safe. And red, yeah, those are way too unstable. Just avoid those completely. And getting this done, it takes like 3 clicks. Seriously, you just go to the graphing icon, pop open your experiment settings, and then you just check the little boxes for the genes you want to test. That's it. The software handles the rest. All right, so one problem down, one to go. With our normalization strategy totally locked in, let's tackle that second hurdle, quality control. And for this, we're going to use the Prime PCR Controls Analysis tool. You can think of it as your own automated QC detective. So what this thing does is it automatically checks the built-in controls, and it's asking 4 super important questions for every single sample. Did the PCR work? Was the reverse transcription step okay? Is there any contaminating DNA? And, maybe most importantly, was my starting RNA any good? It is your complete pre-flight checklist.
But here's where this tool really becomes your best friend in the lab. It doesn't just give you a green light. It also flags potential issues, so you can investigate before you waste a ton of time analyzing bad data. Let's look at a real example. Okay, so check this out. Here's the summary screen from an experiment with 4 different tissue samples. You can see right away, cartilage, heart, and thymus, they're all green. Good to go. But wait, look at that. There's a yellow flag on our skin sample. Let's dig in.
And notice, it doesn't just flash a big red fail sign. That's not helpful. Instead, it tells us exactly where to look. It says to check the reverse transcription control. It's pointing us right to the source of the problem, and that saves us from a total wild goose chase.
So we click on that RT tab to see what's going on, and the software shows us a high delta CQ value. So what does that mean? Well, you can think of delta CQ as kind of like a performance gap between your reaction and a perfect positive control. A small gap is great. A high gap, like the one we see here, is a major red flag. It's the hard data telling us the reverse transcription for that skin sample probably failed, which means pretty much any result from that sample is suspect.
All right, let's zoom out and bring this all together. We've used one tool, to pick absolutely rock-solid reference genes, and we've used a second tool to verify our sample quality. So, what's the big payoff here?
What you're really doing here is building a fortress around your data. It's a two-step validation process that you build right into your workflow. You're not just hoping your data is good anymore, you're actively proving it. You start with a solid foundation, then you confirm every single sample passed its quality check. This isn't just a framework, it's armor for your data.
So here's the bottom line. These incredible validation tools are already built into your software. It just takes a few extra minutes to use them. So the next time you're about to analyze your results, just ask yourself, have I just collected data, or have I built certainty? The answer could be the difference between a good experiment and a great publication.